The cell cycle is a series of events where a parent cell grows and prepares for division in order to form two daughter cells. The majority of the cell cycle is spent within interphase, where it grows and prepares for division. It later moves on to M phase, where it divides and forms two daughter cells. The cell cycle is important because it's the replication of cells which allows multicellular organisms to grow. If cells don't divide, they can become way too big, which makes it harder for those cells to eat and get wasted out, leading to their deaths. Interphase. During the cell cycle, this is where a cell will spend most of its time. Interphase is a period of time where the cell will grow and prepare itself for cell division. Here, it will go through three different stages: G1 phase, S phase, and G2 phase. And it's possible for it to go through G0 phase. There will also be 3 checkpoints during interphase in order to prepare itself for M phase, cell division. G1 phase. During G1 phase, the cell will begin growing in size and will prepare to replicate its DNA. G1 checkpoint. At G1 checkpoint, which is near the end of G1, in order for it to pass this checkpoint, the cell must meet the requirements of being healthy and undamaged. If it doesn't meet any of the criteria, it will go to G0 phase, a resting area. G0 phase. When a cell fails to meet the requirements for the G1 checkpoint, it will remain in G0, where it is no longer a part of the cell cycle. It could also end up here if there aren't enough resources. Here, most of the cells will stay here forever, or until they are needed. S phase. During the S phase, the cell will now replicate its chromosomes. Now it will have 2 complete sets of chromosomes. This phase is also referred to as the synthesis phase. S phase checkpoint. During the S phase checkpoint, DNA is monitored for replication errors. If there are no errors, it will move on to the next phase, G2. G2 phase. At this point in the cell cycle, the cell will continue to grow, and it will make sure that it has all the organelles and other cell structures needed in order to continue with cell division. G2 checkpoint. At this checkpoint, it will be monitored if the cell has no damaged DNA, it has 2 complete sets of chromosomes, and it has all the components needed for cell division. If it meets all this criteria, it will move on to M phase. M phase, mitosis. M phase consists of 4 different parts, which are prophase, metaphase, anaphase, and telophase. During M phase, the cell will now begin to divide. During this phase, it makes sure it has all needed chromosomes and all needed DNA information so that there are 2 identical daughter cells. M phase, prophase. Prophase is the first stage of M phase, mitosis. During prophase, the cell will now have visible chromosomes. The centrioles in animal cells will migrate to the poles. The nuclear membrane that was once in the parent cell will now temporarily disappear, and a spindle is formed near the poles of the cell. Metaphase. Second stage of M phase, mitosis. During metaphase, the chromosomes line up on the equator of the cell, and the spindles begin to attach to the centromeres, which are little things that tie together the chromosomes, the chromatids.
Dashboard phones on the console. Catch you with my cutter, bury you behind the school. Haunted mound, candles lifted when we come around. Scarecrows at the impound. 4 a.m. when we take the town. Cemetery and ghost mound. Sunset across the fairground. Night cry out, no time now. This is Grave House, boys, so we can keep it cover like Nevada.
Anaphase. Third stage of M phase. During anaphase, the sister chromatids that are now attached to the spindle will separate. The centromeres that were containing the chromosomes, the sister chromatids together, will now divide. Sister chromatids move to opposite poles due to the spindles that pull them apart.
Cutter like Nevada, 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 cutter like Nevada. Leave you in Nevada, 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 leave you in Nevada. I've been rocking back and fucking forth in the corner. I've been getting super fucking gone till the morning. You believe in life, you're a dumb motherfucker. If you really wanna try, we will run you over. I've been coughing up blood, dying at McDonald's. I've been mixing Benadryl and tossing in my monster. I'm dead, the world's burning, I believe in.
Telophase, the fourth stage of M phase. Here, just before it moves on to the final stage, the chromosomes will now uncoil. They will now be inside a new nuclear membrane, and we can see that the cytoplasm starts to split, almost split. It has not yet split. The spindle disappears, and it is beginning to prepare itself for cell division.
Monsters, I've been in my grave house, posted with my cutter. I need some glass knuckles, and I need a shotgun.
During telophase, we will have our last and final checkpoint, the M phase checkpoint. During M phase checkpoint, the almost divided cells will have to be checked if they have undamaged DNA, undamaged chromosomes, and if they are healthy and prepared for cell division. If they meet all these requirements, they will move on to the next and final stage of the cell cycle, cytokinesis. Cytokinesis. This will occur at the end of mitosis. Cytokinesis is when the almost split cytoplasm finally splits into 2 new daughter cells from the original parent cell. The organelles are divided, and the 2 daughter cells should be identical to the original parent cell. They will have their own nucleus and their own set of chromosomes and other cell organelles. After reaching cytokinesis, the 2 new daughter cells will go on and repeat the cell cycle over and over again. It repeats.
I'm a skeleton riding out in a black truck. I've been drinking Jaguar and Red Bull and vodka. 100 million dead cops, motherfucker.
In the cell cycle, there are regulators that control the production of the cell cycle. There are 2 genes, the proto-oncogenes, which encourage cell division, and the tumor suppressor genes, which do the opposite and discourage it, or try to stop it. If both of these two genes are mutated, the proto-oncogenes become, and the tumor suppressor genes become inactive. when the proto-oncogenes become oncogenes in uncontrolled division, and since the tumor suppressor genes are inactive, they aren't stopping any of the cell division. This can result in cancer. A quick little visual is this. In class, we learned a healthy cell division in the colon will look like this, where they keep dividing, moving along, and they differentiate over here or anywhere. They just at one point they just die or stop dividing. But a mutated one would start piling up somewhere, anywhere here. The cells would start piling up, and eventually that would lead to a tumor being formed, which would lead to cancer. So basically, these two regulators, these two genes, the proto-oncogene genes and the tumor suppressor genes, they monitor cell division. If both of them are mutated and both of them are defective, it can result in cancer or in any other illnesses. So that's why it's important to keep these two healthy.